All right, guys, welcome back for Unit 3, Discussion 2, Evidence for Evolution. This is going to be a large bulk of this unit and covers kind of some of our main standards here. So we have 5 main sources that we're going to talk about. We have fossils, anatomy, embryology and developmental biology, biogeography, and molecular biology. Fossils should hopefully feel like a bit of a review. We've already talked about them. Remember, they demonstrate a progress or progression of evolution. So we're not looking at individual fossils, we're looking at series of them. We're looking for that change over time and putting things in order for that relative dating. We actually have quite extensive records for whales or horses if you're interested in seeing what this looks like. And then by looking at this overall record, we can see that transition, that intermediary anatomy, and see that change over time. So here you can see a really rough breakdown of how horses have changed in this one specific one. And again, this is artist rendition based on the fossils. Next is anatomy. So anatomy is going to look at shared structures between organisms, specifically homologous structures. And we haven't really discussed homologous yet. We will more later, but these are structures that evolved from a shared ancestor. And the example for this is pentadactyl wings or limbs. You'll notice that humans, dogs, birds, and whales all have that same base structure in their appendages, even though they've evolved and changed to better suit the needs of the organism. Now, don't confuse this with convergent evolution. So, environmental pressures and other factors can lead to two organisms having a similar trait without having that shared ancestor. And we call those analogous structures. So, divergent evolution, which is what typically causes these homologous structures, can also lead to vestigial structures. And these are unused structures without a function that have become obsolete for one reason or another. Third, we have embryology and developmental biology. Now, we're not going to go super in depth into this because it would require a whole class to fully understand. But the base idea is that vertebrates and other groups within their small groups throughout phylogeny do share stages of development, especially in the embryonic phases. Now, this is not based on time elapse. Gestation periods are going to vary from organism to organism, but a lot of the stages are the same. So, a blastocyst for a frog goes through the same stages as one for a chick and a human. So we see those shared developmental steps before they branch into their own unique organisms. Number 4 on our list is biogeography. So biogeography is this idea that the geographic distribution of organisms on the planet follow patterns that are best explained by evolution combined with tectonic plate movement over time. So essentially, the simplest way of explaining why we have the same species across the Earth is that at one time they all started from the same place, and then as the continents moved, they split as well. If you're interested in examples of this, you can check out the marsupial diversification of Australia that was caused by isolation due to it being an island continent. And you can also check out how Protea plants are actually found all over the world in the same climates, despite having no way that they would have transitioned between each other otherwise. Now, I will note this is not covered in the Biozone book that we are using this year, so more information on this will have to be found in other resources for your project. So you can see here we've got the map. These are the Protea flowers as they go across and where they're found. So last but not least on our list is molecular biology. Molecular biology and DNA in general just refers to the shared DNA between organisms and shared proteins as well. So a lot of times the mistake with this one is to think it's only DNA. But we actually have shared proteins and amino acids and things like that that show how closely tied organisms are. So most organisms within any domain, archaea, bacteria, eukarya, etc., share major genetic features within the domain. So a eukarya is going to be similar to a eukarya, but it's not going to be similar to archaea or bacteria or protista, those other domains, because those domains are kind of the dividing line. They were splits that occurred between organisms a very, very long time ago in comparison to the more recent ones. So you'll note that those domains, especially like our components of our ribosomes, composition of our membranes, things like that, those are very early splits to develop those distinct patterns. We can also look at the evolutionary patterns and shift protein sequences. As most mutations occur, they tend to add things and tend to happen mostly on extra copies. We don't typically see changes on critical functions, only because changes that occur are often incompatible with life otherwise. So we can watch for those changes in the sequences and in the proteins, specifically in the less used ones or in the ones that are less critical. So remember, we have this shared ancestry, this genetic code. Everything on Earth, pretty much, that is living uses these instructions for life, encoded into DNA strands using the same type of amino acids. And so this just leads to more support of the idea of evolution as a theory and how we explain all of these similarities and shared features.